at Jawaharlal Nehru University, New Delhi. He is distinguished scientist in field of plant genetics, genomics, plant breeding, and molecular biology. Professor Ramchiary completed BSc from Assam Agriculture University, MSc in genetic and plant breeding from University of Agriculture Science, PhD in genetics from University of Delhi South Campus, 2007. His research focused on the plant genomics, crop improvement, molecular markers, genetic diversity, stress tolerance, and sustainable agriculture. Through advanced molecular techniques, he has contributed significant understanding plant genetic resources and improving crop productivity. His work supports the development of climate resilience crop which is essential for food security and environmental sustainability. We warm welcome to Professor Nirala Ramchiary and express our gratitude for sharing his knowledge, experience, and vision with us. His guide and encouragement will make these sessions highly informative and inspiring the all interns.
Thank you. Done?
Yes, sir.
So, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. What are others? Hmm? बाकी लोग आएंगे नहीं हैं? You'll come, right? No idea? So where you staying? Huh? Narmada. All of you? Sifra. So how you finding JNU? Look sleepy. You didn't sleep? Huh? Huh? Did you sleep or no? You look very sleepy, that's why I'm asking. Huh? How many are from Tesco? 3. What's your name? Ankur, Vijay, Anirban, MBBT, Environmental Science. All of you? Okay. And you're from? Punjab. Environmental Science. Botany. You also Botany? You? Botany. What's your name? Aradhana, you? Rahul. Biochemistry. Same. Your name? Nilesh. That's good. And you can introduce a little bit? Orissa. Which department? Marine biology. You also? Marine biology? Yeah? Yes. Microplastic. Okay. So, visible, right? So I'll be talking on crop plants improvement. Could it be sustainable production of, grains, vegetables, right? Or we try to improve the quality. Quality means textures, nutrient content, vitamins, mineral elements, as well as many other, components of the crop plant. Okay. Those who are from marine and other may not be interesting, but I'll try to make it interesting in simple way. If in between you need to ask anything you don't understand, you are free to interrupt me at any time. Okay? Right. So since my specialization in the crop improvement, I talk about how the earlier village people used to take the seeds from one generation to another generation. Then how it developed to identifying the genetic material that which we say later as a marker, as well as gene as a QTL. Then coming back to the identify gene, then RNA analysis, large scale RNA analysis. We know it's known as the transcriptome. When DNA to RNA, a particular gene produces RNA for transcription, we said it's a RNA or transcript. When you analyze large amount of RNA at any particular tissue, at any particular developmental, you know, timing from any organism, we say it a transcriptome. Right? Clear? Right. Then come to the proteome. We will not go for proteome. Then how the genome sequencing helps to develop crop varieties that are high productive, high environmental sustainable, as well as many other things. Okay. So I don't allow disturbing others. Okay. Try to maintain the time. Right. Whatever it may be, you should try to maintain the time. Right. Okay. Please. Next time onwards. Right. So we'll be discussing about how this evolution of crop improvement, took place from earliest generation to the current, large scale omic is era. So now, if you look at, we always eat, we eat crop plants, but we don't know where it comes from, right? Many of the, nowadays don't want to go for agriculture, right? Agriculture is something traditional, as old practice, which is not lucrative, right? In terms of earning money, in terms of standard of living, what people think these days. Yes or no? Right. But if you, utilize very nice technique, very modern technique, which is very advanced, agriculture can be a very, interesting profession. Right. Nowadays, you might have heard about producing, agricultural products in hydroponics, urban areas, without, this one also, soil also, give the nutrient, you can get. Right. So now, looking back to the origin of agriculture, the Iran-Iraq region is known as the Fertile Crescent earlier. If you look at here, it's the Middle East is known as uh, the origin of domestication of crop plants. Not only crop plants, also the animals. Okay, domesticated animals. From there, it's spread 11,000 years ago. The wheat is found to be earliest domesticated plants. Okay, which people cultivated there because that area seems to be very fertile. Why fertility is required for the crop production? If fertility is there, high production of crop plants will be there, high growing and other things. So eventually after that, subsequently it spread over all over India. Then their domestication happened. Then many of the crop plants domesticated independently, you know in different parts of the world. India is one of the center of origin, Northeast Indo-Burma, that is there, adjoining to China. We get tea, we get rice, and we get many things. Right. Those from Tezpur may be knowing. Northeastern India is one of the like Western Ghats of India. We do have lots of biodiversity in terms of plants, in terms of animals, including the human diversity we get with respect to culture, with respect to look, and with respect to many of the habits. Okay. So once you go northeastern states, if you have not gone, you can find very diversity of people as well. And out of this, we have rice, we have bananas, we have wild citrus, we have sericulture, that insects. All are there, highly diverse in the northeastern states. Right. Coming to here, so now, if you look at, purpose of crop improvement, what are the purpose? High productivity, right? Obviously, it's if there is no high production, farmers will not grow, then we'll be stuck to death, right? If farmers don't produce. This is the purpose one. Second, third, we need to produce those, crop plants which are sustainable in the sense that even though there is abiotic stress, heat, drought nowadays, you know, lots of climate change, we get not good conducive environment. So we have to develop crop plants which are sustainable in production in the harsh environmental condition, as well as there is always found to be evolution of the any organism which attack the plants. If you develop a resistance, then next time what will they try to evolve, mutate in their genome. Then every organism genome has a you know a potential to be a plasticity. You might have heard about genome plasticity. Genome plasticity is nothing but the in the genome there is always we have DNA which helps to adapt to different environmental conditions as well, including that supposing those insects pest they don't get the crop plants uh, you know you know very nice, they will adapt and try to attack again and infest the crop plants, including the microbe. That is what genome plasticity is there. Okay, then we have increased tolerance to herbicide, longer storage period for the harvested crop. There are many crop plants which are not storable for the long time, right? You might have heard about first genetically modified organism, GMO, right? The tomato flavor saver, right? What was it? The shelf life, right? So likewise, is we need to improve the crop plants not only with respect to the one particular trait, multi-direction we have to go for this one, right? So now, if you look at here, why we look? There is chili, this is rice. If you look at this is brassica, this is tomato, this is pumpkin, cucurbita seed. You get this kind of variation. Why? The basic reason we get is because of change in the DNA level, right? Change in the DNA level could be at the change in the amino acids, or could be the nowadays we know the change in the promoter region of the gene. Now, if you define a gene, what is a gene? It's a segment of DNA having promoter, start codon, stop codon, which produces RNA, which it may not be translating or may be translating the protein, but it has a certain function known as the gene. Okay. If somebody asks you what is a gene, okay, you will be able to define a gene. Okay. A, to be functional, a gene has to produce RNA. But it doesn't matter whether it translates into protein or not. But it has a certain function. To be functional, a gene need to produce the RNA through transcription. Clear? Right. And it regulates. Okay. Supposing there is mutation in the promoter region, you might have heard about what is a promoter. It is a DNA sequence upstream of the start codon of the gene. Okay. Right. So when there is promoter, the first RNA polymer is bound to it there, open up, then start the transcription. Clear? When there is change in the promoter region, mutation there will be there. The RNA polymers may not be binding. Some transcription factor may help to bind. Now, coming back to the transcription factor, what are the transcription factors? Those are the genes, okay, that have DNA binding site while interact with the structural genes and helps to express or suppress the gene. Clear? Okay. Now coming back here, so if there is mutation, there will change in the amino acid, change in the expression of genes. We get this kind of lots of variation of the genotype rather than we go for the species. Okay, that is why with the evolution, with the course of evolution, we get accumulation of those mutations in the this one germplasm or different genotypes. We get some may change the color, some may change the phenotype of fruit size, some may change the fruiting habit, flowering habit, or duration like this. Clear? This is very easy to understand. Now this is not visual actually. How the crop breeding started, this is the molecular markers that will come later on. Okay. So we'll come one by one. So this is how it started, crop improvement. This is the 10,000 before present, and coming to 2000, all these things, CRISPR cases come. Okay, what is CRISPR case? I think you will be learning later on, but today I will not be covering. You might have learn little bit in your course, right? This is timeline. What varieties, what people have produced, history of genetic modification of crop plants. Started with the 10,000 years ago, then coming to the again the same, that different crop plants are modified in a such way that through conventional breeding as well as through genetic markers breeding as well as the what through the genome editing or development of transgenic plants. You know what is transgenic plants, right? Okay. So this is not visible. Actually, this is all history only. I can give you later on if you want to. Now, crop breeding. What is a crop breeding? Earlier used to be called as the plant breeding, right? What is plant breeding? It is the branch of agriculture that focuses on manipulating plant heredity to develop new improved plant types for use by society. Unless until you develop a better, genotype, then what happens? Unless you develop a better genotype, there is no point of spending time, wasting time, and developing, new breeding. Okay, it is the development of new and earliest breeding lies in the corn and other. Okay, things. So this is you might have heard about few years back that PepsiCo was filing case against the farmers in Gujarat. Have you heard about that? PepsiCo, this particular potato bred by PepsiCo company, and this is used for production of potato chips. Very nice. There was a huge, debate, and then they went to the court case. That PepsiCo finally court verdict is that, right. This is you can modify the crop plants and patented the crop plants also. You can commercial cultivate also. So you might have heard about Green Revolution, right? 1943, Norman Borlaug is very famous, and unless the wheat developed by Norman Borlaug in Mexico was brought to India, India would have starved by now, right? You might have heard about M.S. Swaminathan, the father of Indian Green Revolution is known as, right? Okay, so what happens is that during the early 60s, because he was the Director General of Indian Council of Agricultural Research, along with the decision of the time Prime Minister Indira Gandhi, discuss and see we have, because you might have heard of Bengal famine earlier. Many people starve, and Indian wheat, okay, varieties are very tall, not giving more grains, lodging, falling down and lodging. But they develop Mexico, Norin 10 of Japanese wheat variety, and cross at Mexican land, they become very highly productive because of that. So bread brought to India, but immediately after bringing to India, it's not survived. Anything that any crop plants from any country you take to other country, firstly you need to have acclimatized it, change the genetic makeup so that it survives to that particular environmental condition. So Indian scientists, cross it to Indian, genotypes, then it becomes, adaptable to environment. That's why, it survives. Okay. Similarly, Erie also developed semi-dwarf wheat rice variety. India brought it, and then India became self-sufficient in grain production. This is one that what he said in Norman Borlaug is, for more than half a century, I have worked with the production of more and better wheat for feeding the hungry world. But what is merely catalyzed a part of the picture. I am interested in total development of human beings. Only by attacking the whole problem can we raise the standard of living for all people in all communities so that they will be able to live decent lives. This is something we want for people on this planet. Norman Borlaug. Okay. This is a great scientist they have, developed. So, how do we improve the crop plants? Future is that strategies, different strategies are there. Identify key traits based on what trait that you want to improve. We cannot do as a whole total. Being left in central universities, you may select some of the topic which is very, very important locally as well as the broader human population also. So there are lots of scope for that. Right. So now, as already discussed, okay, already discussed, this is the first, what was the first identification? Earlier scientists, they have identified natural variation. Look, this is the color different. This is the phenotype different. Let us to take this one. Change from one phenotype to another. Eventually, nowadays, if you go for the hybrid commercial cultivar, almost all are uniform. Okay, what they did is found that this is high yielding, this is maybe quality is good, become slowly uniform genetic material. But if you look at any of the traditional variety, it has different character which are adapted to their particular environmental condition, because they have been mutated, they have been selected, they have been okay, right, then adapted to a particular environmental condition, then slowly multiplied and this one. But nowadays it's very difficult to find local varieties unless few of the research institutes are maintaining. Indian Council of Agricultural Research is maintaining rice and any of the plant genetic, crop plant genetic resources. One is that National Bureau of Plant Genetic Resources, New Delhi. And for rice, we have in Orissa, we have Hyderabad. Sorghum, we have Ikrisar in Hyderabad. There are so many crop plants are being, collected and maintained in this Jamplak. Okay. So finally, if there is no, variation in the crop plants, started with giving mutant, the chemicals, mutagens, then UV treatment, then started looking for the mutation which is different from the original phenotype. Then utilize. If not, nowadays available techniques is that we can modify any of the particular gene segment known as the genome editing. Right, this is the technique here. So what earlier, classical breeding. So now people used to have, how did earlier, because since 1901, crop improvement started. Okay, now they have started with phenotypic trait. Now, when did the DNA was came to know that genetic material? Any idea? Hmm? When was the DNA proved to be a genetic material? 53 only double helix they have given. Before that, 1940, 46. Hmm. Who did that? Right. So before that, what people did in corn in improvement in the USA, they have taken different types of corn trait. They have made a cross and selected against. This is nothing but the mass selection, pure selection, pedigree selection, bulk selection, hybrid breeding. This is without any molecular, molecular technique. Without that also people started doing. Only that after DNA was proved to be genetic material, and then DNA markers developed in early 90s, started with the molecular cropping, and then started the transcriptomic whole genome sequencing after 2000. Okay, now 2013 onwards, we do the what genome editing. Clear, right? Okay. Now have a look, brief look. What are those? Right. This is the mass selection. We have, supposing a farmer collected lots of seed from their cultivation. So he might have found some are seeds are bigger, some are the plants may be growing very vigorously. So what they will do, they select those plants, collect the seeds, and again go for the combine together, go for the next generation. This is nothing but the mass selection. Clear? Means From the lot of variable seeds farmers grow, you will select few of the plants. Okay, then combine together, mix together, grow next year. If it is coming good, then again next year coming good. Then it is basically nothing but a sorting of the good vigor, good quality seed. Because this is the mass selection. What is the farmers can have been doing many years. What happened? Okay, then this is seed selection. You might have heard about Johnson experiment. Random seeds you can select pure line. Then what is the gram? Then you can select the bigger seed size. Then you can cook. This is very simple. Mass selection. You can do the cross. Okay, this is the pedigree selection. Generally, what do we do in the crop improvement scientists? Supposing you have to combine, for example, one disease resistant from one plant, okay, or one quality, good grain quality, another is good, plant types. We have to combine great grain size as well the plant type. Okay, plant type helps in surviving, root may be better. Then grain type, good bigger size. So they make a cross between, but remember that it is within the species of the different genotype. So you make a cross, then develop F1. You know all the Mendel, right? When we cross a two genetically distinct plant, F1 will be highly heterozygous. From F2 onwards, they will be segregating. Okay, segregating means any part of the genome of parent one can combine with any part of the genome of the parent two in the progeny. This is because it happens because of there is meiosis formation. What is meiosis? The recombination takes place in the during the meiosis. You know the cell division, right? In science, mitosis and meiosis. The moment you allow to meiosis to happen, there will be recombination of the genome of the parental line. That's why we get the phenotype. And after F2, every plant's performance is recorded in pedigree. Means this is F2-1, F2-2, F2-3, F2-4, something like that. So how it is performing, what is the seed size, how it is plant type, that will be recorded, and the last year all the good performing plants are combined together and you can release it. Clear? Pedigree means after F2 onwards, performance, seed size and everything will be recorded, keeping in your head. After that, we can do it. Okay. So this is backcrossing. You know what is backcrossing, right? Backcross means P1, P2, then any of the parental line with backcross. Either it could be transferring particular, for example, basmati rice. You take basmati rice, very grain quality and something, but it doesn't have resistance to a particular disease. You want to combine that resistance gene of any of the particular rice variety along with the good quality of the basmati, right? So what we will do? Make a cross with the basmati and the resistant line. Then again make a cross again and again with the basmati, so that except the gene of resistance, all the genome of the basmati comes back to the progeny. This is known as the backcrossing. Clear? This is very easy. You might have learned this one. Backcross breeding. Then we have bulk method. Bulk method also. You just harvest, grow it, harvest it, seed, and bulk. Again, grow it, harvest it, bulk, grow it, harvest it, bulk. And after 8 to 6 to 7 years, you will get the new berry. It is nothing but a purification of the removing of the unwanted seeds which are lowly performing. Right. Single seed descent method. What happens? From every F2 plants, you collect only one seed and keep the other seed there and then grow. You have record. Okay. Supposing you have F1 to F2, you get 200 seeds. Out of 200 seeds, F2 will produce hundreds of seeds. No? Every F2 will produce hundreds of seeds every plant. You select only one seed, grow again. Again, next generation, making a cross, grow the plants, making a cross, see the F1 in performance. Then again select, again you can go for the selection again. Here is the recurrent selection is that nothing, same type of selection we can do again and again by making cross. Why making a cross? Just to see that how better performance, because combination of different genome from different genotype will give more performance of the plants. Okay, right. So this is reciprocal recurrent selection. Take pollen from one plant to another side, and this side to another side, grow individual lines in the plot. Then you go for the improvement. Look for this one. Okay, so you can study this one. Now coming to the recent one. Earlier one is not so. I feel it is very easy, right, for you guys, and nobody is interested to it. Now coming to a little bit molecular technique, right? Okay. When you say molecular, the current generation Gen Z is much more interested than going back to the phenotype solution, right? So now, as I already told you, 1980s, late 1980s, first DNA markers was developed in tomato, 1987 or 88, something like that. Then started, you know, using DNA marker. Now coming back to the what is a marker? What do we say a marker? A marker is nothing but it creates distinctness from one individual to another individual. Okay. For example, if you take a book. Okay. On the book you have read many sentences. Okay. You'll mark with a pencil. Okay, marker something differently. So it creates. This is the unique. This is you are marking with a marker. It's a marker, right? In plants also, we can have a marker. For example, flower color. One is red, another is what? Yellow, and another is blue, something like. Right. But problem to be marker is that full genome. Genome is nothing but what is a genome, right? Yeah. Genome. What is a genome? Known as genome, right? What is genome? Hmm? Hmm. Now let me clear your concept. In diploid, haploid complement of chromosomal DNA is known as the genome. Why? Same copy. Okay, right. When you say that, for example, Arabidopsis is a plant, it has 10 chromosomes. When you sequence, we always get the 5 chromosomal genomic information. It is a sum total of DNA present in a haploid cell. Haploid, right? Genome. Now, how many genomes are found in the plants? How many genomes are found in the animals? Can anyone tell? Where do we get the genome in plants? Hmm? Hmm. Nuclear. One more. Mitochondria. Right. So if you say genome, means it's a DNA complement. We find 3 genomes in plants: nuclear, chloroplast, and a mitochondria. But animals we get in the only mitochondria and nuclear genome. Right. So now, what happened? I think ask for password. Huh? Huh? Star 18. Huh? Marker, right? Which creates or mark differences from one among the another. Right? It could be phenotype, but genome size is too big. For example, human being has a how much? 22, 21 plus 1 plus 1. Right? Okay? So 21 is autosomal chromosome X and Y, we get 21 plus 1 plus 1. Right. So now, if you take flower color, it may be only one gene finally, but inside the plants or inside the animal, any biosynthetic, any metabolism is governed by not only single gene, it is many genes. Okay. But final product, if you associate in final genes, you may get the final product of the entire biosynthetic pathway, for example, color. Right. So it doesn't cover the total, chromosomal DNA. Right. It can be used marker, but a marker has to be unique in every part of the chromosome. For example, human being 21 plus 1 plus 1. We need to demarcate every part of the chromosomal DNA uniquely with different DNAs. Okay. So that phenotypic marker which observed like flower color is not, covering total. Secondly, the flowering time, for example, if you want to get a flower, color, flowering time has to come. We have to wait until it flowers. Thirdly, it may influence by environment. If environment is not good, that red color may be little pinkish or something like it may change. Because of that, people are looking into different markers which can be used without being affected by the environmental condition, which is which can cover the total genome, okay, which is not tissue specific, growth developmental specific. Then came to the enzymes. Enzyme also same. For example, we have 20,000 genes in our body, right? After 20,000, all genes will not be expressed in any of the tissue, except for some of the housekeeping genes. Some genes may be expressing in finger, some may be in ear, some in the eyes, and some may be, right? So if you take the protein or enzyme markers, it will be tissue specific. Again, it will be affected by the environmental condition, and it cannot cover the total gene. Then explore the DNA markers. DNA markers is present in the total genome. DNA is present in total genome, right? And it doesn't get affected by the any environmental conditions. And irrespective of tissue you take, DNA will be the same. That's why I came to the DNA was known as a molecular markers. Okay, using these DNA markers, you can go on. Now, marker-assisted breeding, selection, speed breeding, double haploid, tiling genome, and many more, technology came. Right. So this is how improvement of crop plants 1990s started phenotype-based selection. Then coming to the 2020, we have next generation sequencing, so crop improvement. So now coming to the when there is marker, how do you know that which DNA marker is present in which chromosomal DNA? Okay, and always based on the recombination. What is recombination? Make a cross, and then based on the gene recombination, we get the distance. It is the relative distance of the genes or DNA markers between organisms which will be coming. So why genetic mapping is done? To study the structure and organization of genome. Number one, find the functional genes governing traits of interest. Okay, in the plant genome. Comparative evolutionary studies. Genetic map helps in positioning of sequence context in proper place in chromosome. These are some of the important application of construction of genetic map in any of the organism. Clear? Okay. Now, finally, if you map, why it is mapping? Why it is mapping? Just because we need to identify chromosomal location where we get, genes governing important trait. For example, vegetation, for example, grain quality, for example, grain size. Right. So now there are different mapping technique. One is genome mapping. Genetic mapping is nothing but a relative allocation of the markers in the chromosomal DNA based on the recombination. Okay. Physical mapping, there physical mapping means actual mapping, restriction mapping, cytogenic mapping, fluorescence to hybridization. We have mapping through whole genome sequencing. These are the some of the different technique used for the dissecting the genome of any organism, especially in the plants. What is genetic mapping? Uses genetic techniques to construct maps showing the position of genes and other sequence features on a genome. Physical map is the actual map, actual distance we get. And remember that which one is the actual map out of physical and genetic, because genetic may change based on the number of population you take to calculate the recombination frequency. And recombination happens in the meiosis of the sexually reproducing organism. Clear? Okay. So now what do you require for genetic mapping? One is segregating population or population with known relationships, polymorphic markers, trait phenotype scored in population for QTL mapping. So now coming to the segregating population, will be coming. What are the disagreeables? What is polymorphic markers? The markers should differentiate between one individual to other individual. This is the polymorphic. Okay. What is polymorphic? The markers which we use as a DNA marker should differentiate one individual from the other individual. Unless there is polymorphic or different morphology of the markers, we cannot use for the genetic mapping. Right. So what do you need? Then QTL mapping also. So what do you see when you make a cross? You P1, P2. Okay. Then what happens? For example, with respect to one chromosome, one genome is represented, one parental is represented dark color, another is light color. If you look F2 generation, you will get recombination, highly heterozygous. Okay. If you look at F2 generation, you will get highly heterozygous. But if you go there, you get the homozygous combination of both the genome of both the parents. Okay. So now you may ask why we cannot maintain the genetic constitution of F2 and heterozygous population, because the moment you allow the plants to produce gametes, there will be suppling of the genome of the parental genotype. That's why there is reconstitution, recombination, and formation of new genetic combination. Clear? This is how different populations are there. Becross, you have double haploid, and homozygous lines is required because to when you need to sequence or you need to grow the plants known as the immortal population, which is recombinant inbred lines. And I'm sure that you might have heard about pollen culture, enter culture. When you culture, you can have double haploid and highly homozygous plants. If this is the recombination, you can see if you have. I'm going a little quick. With respect to one chromosome, these are a different population of the Arabidopsis. If you see the different parental genome, you get lots of recombination like this combination. This one. Okay, that is what marker genetic map can be done. So markers, there are different markers, DNA markers. Okay, the advantage of DNA markers I have already told. Phenotypic or morphological, molecular, cytological, and all the different markers you can read later on. But I'll go for the only few one. Right, these are different markers. Now coming to the very simple markers. Okay, this is known as the simple sequence repeat or microsatellite. What are those? We get repetition of nucleotide in the genome, plants or animals, higher periods. Like for example, GT system like dinucleotide. We can have CTG trinucleotide. We have ACTC something like polynucleotide and pentanucleotide. So why it is different from one individual to another individual? We get abundantly different between individuals. The flanking this repeat sequences. For example, one individual we may have this tetranucleotide 20 times repeat, another individual you may get what? 10 times repeat. So we get the, when you amplify the DNA flanking these SSR markers, we get differences of DNA when you run in the gel. This is known as the polymorphism. This is widely used because it can discriminate between the heterozygous and homozygous individuals. Okay, crop breeding. So now this is the one that with figure I am showing you primary division flanking. One sequence is like this on one individual, another individual like this. If you amplify, we get differences, right? So this is the amplification. We can see differences of the SSR primer. So this is recombination. Now, if you map, the genome, we can have this like a chromosome. This is the cytogenetic fish mapping. You might have heard about fluorescence in situ hybridization, where you fix the chromosomal DNA and level the, your probe of DNA sequences, nuclear sequences, a fluorescence dye. And since DNA is a, complementary single-stranded DNA and level probe, when you hybridize together, based on the complementary, it will go to the region of complementarity and give the color. Okay, and sequence, if sequence we can have, genetic map can help in the, perfect assembly of the DNA sequence when you sequence it. Now coming to the trait, you might have heard about quantitative and quality. Quality means flower, red or white, right? Quantity means how much red, dark red, light red, medium red, white. There will be in terms of quantity you can get, right? On quantity has always unit like centimeter, gram, okay, right? Then in terms of concentration, in terms of number, unit. Qualitative, we don't have, numbers, just visual, no unit, okay. Qualitative are, highly heritable in the sense that only those traits which are highly heritable means highly, expressing can be scored as qualitative. But those traits, agriculturally important, for example, human height, for example, can be scored as the quantitative trait with the unit. Clear? This is very simple, right? Now, coming to the actual, how do we use markers for the identification of chromosomal region, you know, governing particularly. Now, you might have heard about QTL, right? Right? QTL, what is it? So, how do you define it? Anyone? A region of chromosome flanked by molecular markers which is found to be associated to govern a particular phenotypic trait, quantitative trait, is known as the QTL. Remember that a QTL is a chunk of chromosome, segment of chromosome. It may contain many hundreds of genes. Okay, hundreds of genes not necessarily governing the trait of importance that you are looking for. There are genes which may govern other traits also. That is why we say it is a quantitative trait locus. And when you say quantitative trait locus, you need to always associate with the molecular markers. Okay, if no molecular markers, no quantitative trait locus. Okay, the moment when you say gene, when you say the quantitative locus, when you identify sequence of the gene, structure of the gene, then we say it is a gene. What type of gene is there? For example, structural genes, transcription factors, or something like that. When you say QTL, in QTL we don't know the gene yet. We are just associating a chromosomal DNA of any organism with particular phenotypic trait. That's why through the molecular marker, that's why we say the QTL. Now clear? What is the difference between QTL? It's nothing but the chromosomal DNA associated with markers which shown to be governing, influencing a quantitative trait. Okay, clear? Now, if you look, how do you identify a marker and a trait? For example, this is from the same chromosomal DNA marker, and you know how to use PCR, right? Always we need to have two primer together. So earlier we discussed about, for example, SSR. One individual we have 20 repeats of 4 nucleotide, another individual 10 repeats of nucleotide, but using the same primer. Clear? Look at the down, A and small a. For example, A is from the one parent, small a is from the another parent. Right? Now we amplify these two, the same primer combination. You get one in the down, other in the up, because of the whenever there is less repeat, we get the larger migration. Length is smaller. Whenever there is more repeat, we get the larger DNA band. Okay, upper band. Now, how to associate a marker with the marker with the trait? For example, this is a leaf. It's like thorn, like small structure known as the trichome. Okay, trichome is nothing but a thorn-like structure, small trunk. If upper band is there, homozygous, we get more number of trichome on the leaf. If lower band is there, very less number of trichome, almost negligible. But if in medium heterozygous, we get the medium trichome. This is how we associate. Means this marker seems to be associated with the trichome trait, and this marker can be used for the what manipulation of the trichome trait in the plants. If you look at the genome, for example, the blue is having the high trichome, the red is having the small trichome. You can associate in the quantitative trait locus mapping. Clear here? Clear? DNA when how it is associated? Associate based on the the trait correlating with the genetic constitution, which is known as the genotype. Now, QTL, there are different QTL mapping methods. Single marker analysis. We have interval mapping, composite interval mapping, different, then multiple interval mapping. I'll finish in 10 minutes, okay, 10-15 minutes more. It's fine. Right, I'll try. So what happens is single marker, for example, you have map marker. Look at here. This is the one marker here. So here, when you associate statistically, the QTL may be this side or this side. Okay, this is a single marker. And if this is far away from the main actual QTL, it may give very weak signal. This is the disadvantage. Interval mapping, what happened? There are different marker, one marker combination, many markers in this chromosome. At a time, two markers we analyze associated statistically with the trait. That's why interval mapping. But problem is that supposing this is the two marker interval. This is one interval, this is another interval. Means three markers are flanking, two region, right? So when you analyze, association of this chromosomal region with this marker, the other, position may influence here. So you may never get perfect marker. That's why composite interval mapping, multiple interval mapping, what they do, when you analyze that association of this chromosomal DNA with phenotype, you take control of another one. So it get more accurate analysis of the marker. Clear? You can read this one. So now association of this one. For example, here left figure and right figure. If there is differences in the phenotype along with the marker, we say the association. If there is no differences between A and B individuals, the right marker X marker, then it is not associating marker. So if you look at here, for example, we get that gel band also, that whenever there is association, we get this kind of marker. So for example, we can have this kind of extreme phenotype. For example, you make a cross between resistant susceptible individuals, and in the F2, suppose 200 F2 you get. Extreme means highly resistant, highly susceptible. You take the DNA and do the marker analysis like this. Whenever there is exactly equal. For example, bulk segregation analysis, what you do? For example, P1 and P2. P1 is susceptible, P2 is resistant. Right? Make a cross F1, then go to F2. Then F2 there will be 200 individuals. Now you get 10 extreme plants where highly resistant, 10 highly susceptible. You extract individual DNA of resistant, susceptible 10, 10, and parental line P1, P2. and bulk together of resistant, all the 10 resistant, all the 10 equal quantity of DNA, do the marker analysis. If 10 resistant is found to be DNA when is equal to the resistant parent, then we said it is the matching. Okay. If susceptible, DNA when is matching to the susceptible, F2 progenies, we said it matching. For example, if you look the cursor, this is P1 and bulk 1 and P2 and the bulk 2, you get there is, matching, right? So this way you can screen the susceptible resistant. Can you get what I am telling? Hmm? For example, this is susceptible, this is resistant, right? Okay. Now you take bulk 1, bulk 2, extract the DNA of this. Individually, this also DNA of this. And parent 1, for example, it is a susceptible, parent 2 is the resistant. Now, if take the marker, for example, this is P1 and all susceptible plants are same DNA, this arrow, and resistant P2 and all the DNA of this resistant, resistant disease resistant is same, means this marker is associating with the disease resistant. Clear? Okay. So now this we represent like this in QTL. These are markers chromosome. This you can represent like this. Okay. Now how to select backcross? For example, F2 plants. For example, P1 susceptible, P2 resistant, F1, F2. You get lots of plants without looking for the infection resistant line. We get through the DNA seedling stage. This DNA markers resistant, susceptible. We can select those plants. Arrow 1. Okay, and again look for the backcrossing. Clear? Whenever there is DNA marker linked to the resistant phenotype is there, you can select it, again take to the next generation. This is how it is done. Now, based on this, when I was in South Korea, this is known as the club root resistant. Okay, this is the resistant. When you make a cross and map, we found there is one gene, associated with the club root resistant in the progeny of susceptible and resistant lines. Then we map and found the S and B markers. Similarly, for example, this is the diamondback mud, highly, this is diamondback highly, infesting with its cabbage plants. So there was resistant one line, another is susceptible lines. So we did analyze and found that resistant line one, this is the marker. All the susceptible lines, this is a marker, and this is being used by the company for breeding diamondback moth resistant cabbage lines. Similarly, that cabbage we have known as the cabbage is on a head. Okay. Without maturity, before maturity, sometimes whatever, or when it is matured, it breaks down, known as splitting. So based on the resistant and susceptible of splitting, we develop markers. This is being used for the breeding resistant lines. So line. Now coming to the next. What is now molecular biology? Reverse breeding. Have you heard about reverse breeding? Suppose company people have given you one F1 line. Now it is F1 line, hybrid lines. Company sell you. We don't know the parent line. Okay. Now how to develop? Scientists have developed how to develop same hybrid in your laboratory. Can you get? Because company is not giving, because it is patented. So what happens is they have found some of the genes which help in the recombination. Okay, genes which helps in recombination, because F1 F1 is one from one parent, another other parent combination. There is no recombination. So when you apply the silence the recombinant, the genes which is helping in a recombination between two genome of the two P1 and P2, there will be no recombination of the genome. Clear? You know that when the meiosis happens, there will be recombination of the genome. Right? Clear? Now, this plant, what happened? Commercial hybrid, variety you got. F1 hybrid. This is not recombinant one. The moment you allow to meiosis, chance is that there will be recombination. Take the plant and do the silencing of the or editing of that genes which help the recombination. Then what will happen? There will be no recombination. Chromosome as a whole will segregate. Clear? So after that, what will happen? You get the individuals. For example, here this is recombination. Also, you can get so individual which is not recombining. This is the individual. Total chromosome is going there. Total chromosome is not going there. So if you make a cross between these individuals, then what will happen? Look for the phenotype of F1. Now remember, first F1 commercial cultivar we get. We did the genome editing or genetic engineering so that recombinant gene is silenced. When recombinant gene is silenced, there will be no chromosomal recombination. So what will happen? Then chromosome will be inheriting together. Now take those individual plants, try to make a cross between the plants that you get, and if you F1 you get the same individual as hybrid, means you have produced the parental line. This is nothing but the reverse splitting. Okay, right. So this is the one, and same is there. Now split breeding. What is split breeding? It is nothing but the, for example, in natural environmental condition, what we will do? Natural environmental condition, we grow one season in a year, right? Split breeding, what happened? What do we do? We can give the temperature and the light. This is stress condition. You can multiply. For example, this is wheat, barley, and other thing. You can multiply. For example, natural condition, you can have 2, here 4, 3, here 6, something like. You can basically it is a artificial condition when you get more stress, multiply the seeds, and advance the generation many generations. Then within 2 years you can, instead of 10 years, you can get the plants. Clear? This is the one. So now coming to little bit 5 to 10 minutes. What is omics now? Right? In omics is large scale analysis of what? DNA, RNA, protein, metabolite. We say it is a omics. So high throughput is a process. For example, DNA sequencing, high throughput sequencing, RNA sequencing, high throughput sequencing. What are those? Next generation sequencing, known as the second generation sequencing, and the third generation sequencing, known as the long read single molecular real time sequencing. What are the differences? Next generation sequencing produces highly accurate but shorter DNA sequence. Third generation sequencing you might have read. Mukesh has covered yesterday. Right? So no need to go. Right? So now just basically what we need to cover is understand is that when DNA produces transcript produces RNA known as transcriptome, analysis of the RNA of any particular tissue from any developmental stage of any organism known as the transcriptome. We have proteome, like if you analyze the proteome, then it helps to produce the metabolites, which metabolites again helps to produce the what exactly will like known as the phenotype. Okay, for example, different colors of this one. So what we did is this first generation, third generation, you might have already read. Considering that Mukesh has covered this one, insertion of addition, the one which we see the blank, we see it is a deletion sequences known as the indel markers. Now, coming this concept, I'm working with the capsicum known as the chili peppers. But generally, when you say the capsicum, we understand that this is only Simla mirs bell pepper. But all the chili species belongs to the capsicum genus. You may remember the binomial nomenclature. Right. So all the genome contains the genus name is the capsicum species different. Now you might have seen the Better India. Better India is very informative in the sense that it gives cake map of India, pickle map of India, chili map of India, any map of India. Right? If you go to Better India, I happen to come to this picture, and if you look at, there are different, chili grown in different parts of the India, out of which that northeastern states has 4 5 different unique chili species known as the ghost chili and the Dale Kursan, this one, Sikkim, the highest pungency containing chili species in the world earlier 2007 reported. This GI tag has come, different. This is GI tag chili peppers. So now chili is, measured. Chili is unique because of it has pungency, right? Known as the capsaicinoids. Okay, spiciness. This chili in 2007, notice testful, they'll know. Testful itself, DRL reported highest pungency containing chili in the world. Immediately after, DRDO reported, all the Western people came to India and took away all this. Developed so many products, okay, out of this. This is, this is common chili down, less pungency. This is our, and using this genetic material, New Mexico State University has developed the highest pungency now, more than 2 million Scoville unit. The pungency is measured in terms of Scoville unit. The person who developed the measurement of pungency known as the was Scoville. So ghost chili. So now, if you look at ghost chili Bujulukia, there are lots of cartoons like this with so many different dangerous cartoons. Have you seen that? There's one movie when the person rides the bike, all burn down. Ghost Rider. Many people, compare this Ghost Chili with the Ghost Rider. Right? Okay. Now everywhere it is adapts. It's very interesting because when I was, South Korea in 2008, 11 to 11, I wanted to see if anyone will work on this, but nobody worked. So in our trial, we also try to see that, you know, many people in Northeastern they use for the when you have fever, give soup, chili soup, some chicken with adding chicken or curry, soup, fever and all this. You drink it, full of body sweating, every fever goes. This is traditional medicine. We tried. If you look at that, if you carcinogen, benzopyrin, cancer tumor is, more. But if you extract of this ghost chili, Bojolokia, very small tumor size and all this. Recently it came and we published this paper and published in notice in paper in a newspaper Times of India, all these things also. It's very, productive. We found that is, chili, stomach as well as lung cancer. It is reducing tumor in size. So now it has many old records. 2009, when Gordon Ramsay, you may be named, famous UK cook. When he came to India, this Aninita created world record. And in 2000, this one, Jack Ma, Mike Jack, in Canada created world record by eating more number of chilies in 2 minutes. Okay, many more old records. This fellow tried to create world record, but they had a, hole in esophagus. He has to hospitalized. Again, US one fellow created world record recently. And if you want to try some different way, you can also create a one world record. Right? You can try it in if you want. There are so many students from textbook. You can ask the material from there. Right? So, if you remember Nobel Prize winner, the sensory how it is signals transforming, and David Julius, University of California, he got Nobel Prize for using capsaicin how to, do this one. So DRDO has been using this for development of chili grenades, and many of the Assam police using. And this is 2015. There's a good example that one terrorist from Pakistan came through Kashmir. He was fighting the Indian army, and then with AK-47, Indian army again fire back with chili grenade of ghost chili. After 5 to 10 minutes, they found that no firing came back from the terrorist side. They went inside and they reported that he was literally crying. All the tears came because one chili I grind here will be sneezing here. I mean, tear comes. So ultimately he was caught by Indian Army. So this is where commercial products, before we utilize our own material for our commercial cultivation benefit, people have used in UK and US and Japan. There's so many, the commercial product. Till then, we are not using this one. But at time 2007, it became world famous. They're using this one. So vodka also, and then now it is so many products, and then Ayurvedic formulations, and then cocoa also mixing. So what we're looking is that we're looking what is the pungency, okay, through genomics, many omics, color, and the aroma content. So we have developed, cultivated many of the, chili. Then we found lots of variation in the flowering, fruiting, and then pungency content, food size, and other thing. Then pungency is composed of 14 alkaloids, out of which these are measured. Capsaicin is almost 70%, dihydrocapsaicin is 20%. So it's analyzed and did as the CMS analysis, we find variation in the ghost chili also, and capsicum fruitation small chili. We find, then we did transcriptome analysis to understand how many genes are involved between different, there's a medium pungent, this is low pungent, this is high pungent. We did expression analysis. We find that there are many genes which are expressed very highly in the high pungent, medium pungent. So we have validated through QRT-PCR analysis and found that all the genes are highly expressed in the ghost chili compared to low pungent capsicum annum. So it is high pungent chili. Again, we try to see that because one gene contains more than one copy, and we found that analysis found that more than one copy gene is expressed in ghost chili, pungency biosynthesis. That's how we get more pungency analysis. So ultimately, what we dissected is that through the expression and sequence analysis, we got this developed molecular markers which can be manipulated in the desired amount of uh, desired amount of uh, desired amount of pungency. This is the markers we develop. Genomic part I have not covered. Okay, we did lots of whole genome sequencing also, transcriptome analysis. So, seeing the expression analysis of genes found to be highly expressed in the high pungent compared to low pungent, medium pungent. Finally, we developed 14 gene-based markers which can be used for the manipulation of pungency, like ghost chili and the chili plant. So thank you. I stop here. I see you people are going to be sleeping after some time. I can understand because these are the people who work with me and lots of work for the last 13 years. My students who are placed in different university institutes now. Okay, some are in US doing postdoc. One is open a company, and then they are in different jobs now. And the current left members, master student and then PhD students. This is our School of Life Sciences. Come sometimes if you want to come. School of Life Sciences. Right. Thank you. Anything that you want to ask, you can. Actually, this whole is for at least 3 class. Could you get something to this lecture? No? Yeah, anything you want to ask? If you want, I will send this later on. Okay. Slide. Information. Provided you feel it is important. Huh? Nice picture. Huh? So anything you want to know? No. थैंक यू। समझ में नहीं आया? हैं? हैं? मेरीन बायोलॉजी लिटिल बिट डिफिकल्ट ना, बोटनी में तो थोड़ा समझ में आएंगे। यस। हैं? Speed. Speed you make a, like you might have seen the growth chamber ना, greenhouse. Greenhouse देखा है? देखा कि नहीं greenhouse? Anyone? नहीं तो SLS में आ जाओ। तो it's like a control environmental condition. Okay. आपने सारा इसमें पौधा ग्रो करने का ये है। So when you give stress, lot of high temperature, okay, little drought, the plants become more stressed and they become more flowering, early flowering. सपोजिंग आपको 6 महीने चाहिए तो वो 2-3 महीने में आपको फ्लावर दे देंगे। तो स्पीड ब्रीडिंग इट हेल्प्स इन रिड्यूसिंग द टाइम फ्रेम। आपको 6 महीने यू आर रिड्यूसिंग टू 2 months or 3 months by giving the environmental condition which helps to flower fruit, and that fruit again you take back little. See, it may be so healthy, but as long as you go again and again, you will get more healthy. This is speed breeding. Nowadays, people are using this one. All are, very nice speed breeding facility. You advance designation. Hmm. Hmm. जीन पे there are actually slightest environmental fluctuation में आपका जीन fluctuate हो सकता है. That's why many of the experimental result you get in lab may not be reproducing in the field, right? So we have many mechanisms like थोड़ा सा चेंज होने एपिजेनिक फिनोमेना आ जाता है. You might have heard of epigenome, right? Epigenome क्या होता है? When there is methyl group in the cytosine residue, when you wait at a promoter region, the gene may not be expressed or may be expressing very less. So it helps it change. But as per possible, we try to give the normal condition in the greenhouse. But when you go to field condition, it may slightly differ or drastically differ. That is a problem. There will be always differences in the natural condition compared to the normal greenhouse growth chambers. Okay. So anything else? कुछ नहीं? और भी लेक्चर है या नहीं? हाँ. Nowadays हमारा इंडिया में बहुत सारे लेबर्स है ना, लेबर्स मिलते हैं. If you go to Canada, America or Western countries, they don't have labor force. So what they do is that हमारा means unwanted plants that are present when you grow the crop, right? So what they have developed, they inserted gene in the crop plant, and they spray the herbicide which kills the plants, unwanted plants in the field. So those crop plants having the gene will not be affected, but those not having the gene will be affected with the chemical. Okay, that's why they have developed so that the wheat unwanted plants can be removed from the field condition without employing labor. They have developed a transgenic, inserted a gene. When you spray the a chemical, crop plants will die, but all other plants will die. This is the herbicide tolerance. Clear? Hmm. Not necessarily that there are many studies. People say it is affecting, but till now, आप जो सोयाबीन ऑयल खाता है, सनफ्लावर ऑयल खाता है, और मस्टर्ड इंपोर्टेड जो है कैनोला ऑयल, all are transgenics. हम खा रहे है ना. Now if you look at some of the this one agrobacterium transformation में हम बोलते हैं कि ये agrobacterium TTNA may not be, or the antibiotic resistance gene may not be good for us or something like that. Okay, marker gene, but not knowingly unknowingly we are eating inside. accelerator plants, but it's not happening. Right. There is a major concept is that Europe also they are not growing the transgenic plants, but they are all importing the oils, the food grains. They are also not affecting now. There are some study where they showed may affect, but only problem is that when you develop transgenic plants, which is super, it may produce super plant, right? Super plant cross pollination हो जाएगा, तो आपका marker gene हो जाएंगे, which will not be good for the environment. There are debate. India में भी हमारा जो India develop brinjal GMO, transgenic. Bangladesh is going earning lots of money and importing. We are importing Bangladesh Bengal. Huh? It's happening actually. No? Bangladesh also now producing lots of this brinjal. They're eating that one. So there are reports where few may show that negative, but majority of the publication they show it's not you know uh, negative impact on health. And India, that who oppose the transgenic, they're mostly not from the science background. Hmm? If you Greenpeace or something like, there are so many social activists who opposes this one. India is importing lots of oil. India has developed transgenic mustard, but India is not allowing to grow the transgenic mustard. But we are importing soya oil, corn oil, canola oil, all the transgenic. There are different policy making, problem actually. That is it. Okay, all the best. If you want to see that, SLS, you can come. Anyone in SLS? Which lab? I see. Govind ji. दो ही है चलो थैंक यू डैन for your patience